In this video tutorial, I am going to discuss about urea cycle. What is urea cycle? Urea cycle is the cyclical process by which toxic ammonia is converted to less toxic and more water soluble compound, that is urea. This urea cycle occurs exclusively in the liver. Once urea is formed by the liver, it enters into the blood and goes to the kidney. And from the kidney, this urea gets excreted in the urine. Now, as we know that urea cycle occurs in the liver, but if we talk about the cellular level, urea cycle occurs partly in the mitochondria and partly in the cytosol. Urea cycle itself is a 5-step process. So, first 2 steps, that is first and second step, they occur in the mitochondria, and remaining step, that are 3, 4 and 5th step, occur in the cytosol. Now, urea cycle is not unique in a way that it occurs partly in the mitochondria and partly in the cytosol. There are other cycles also which occur partly in the cytosol and partly in the mitochondria. For example, we have gluconeogenesis and heme synthesis. This both pathway along with urea cycle occurs partly in the mitochondria and partly in the cytosol. Now, if we talk about the history of urea cycle holds very unique position in the history of biochemistry, because urea cycle was the first ever cycle described. It was first described in the year 1932 by 2 scientists named as Hans Krebs and Kurt Hensleit. And in honor of these 2 scientists, this cycle is also known as Krebs-Hensleit cycle. Krebs-Hensleit cycle. Now, after the first discovery of this urea cycle, later on its all steps were described in detail by 2 more scientists, and their name are Sarah Ratner and Philip Cohen. So, these 4 scientists had given their important contribution in the discovery of urea cycle. Now, before looking at the steps of urea cycle, it is wise to first look at the structure of urea. So, in the structure of urea, there is central carbon bound with the oxygen, and on both sides of this carbon, amino groups are bound. So, one amino over here and another amino over here. As we will see in a short while, that this one amino group is donated by the free ammonia, whereas another amino group is contributed by the aspartate, whereas this central carbon along with the oxygen is contributed by the bicarbonate, and bicarbonate themselves are generated from the carbon dioxide, right. So, these are the contributors of various skeleton of this urea. Now, let us look at the urea cycle reactions. So, as I told you that urea cycle occurs partly in the mitochondria and partly in the cytosol. So, first 2 steps of urea cycle occurs inside the mitochondrial matrix. In the first reaction, bicarbonate reacts with ammonia, and in this reaction, bicarbonate ammonia, free ammonia reacts and generate carbamoyl phosphate. Now, this step, it requires energy. Basically, it requires 2 ATP, but both ATP's role is different. One ATP is considered as a substrate because one high energy bond is broken and ADP is released, whereas the inorganic phosphate gets incorporated into the product, that is carbamoyl phosphate. So, this ATP can be considered as a substrate of this reaction, whereas one more ATP is used, which is used for the provision of energy. So, it is one high energy phosphate bond broken down and gets converted to ADP, and inorganic phosphate gets released. This step is catalyzed by enzyme carbamoyl phosphate synthetase 1. So, enzyme is carbamoyl phosphate synthetase 1, which is often abbreviated as a CPS1. This enzyme is of the ligase class of enzyme. One similar named enzyme also occurs in the cytosol. In the cytosol, that enzyme is called as a carbamoyl phosphate synthetase 2. This carbamoyl phosphate synthetase 2 is vastly different from this carbamoyl phosphatase synthetase 1. This CPS2, it is basically, one striking difference is that this is found in the cytosol, whereas this CPS1 is found in the mitochondria. The second difference is that this CPS2, it is required for the pyrimidine synthesis. Pyrimidine synthesis is the cytosolic process, and the CPS2 is located in the cytosol, so it will help in the pyrimidine synthesis. Here, as we can see that CPS1 is utilizing free ammonia as a nitrogen, whereas in case of CPS2, instead of free ammonia, this utilizes glutamine. So, these are the difference between CPS1 and CPS2. And yes, one more difference is that this CPS1, it requires one allosteric activator, that is N-acetyl glutamate. Now, N-acetyl glutamate is often abbreviated as a NAG. Remember, this N-acetyl glutamate is the allosteric activator of the carbamoyl phosphate synthetase 1, and this is compulsory requirement. This is compulsory because without N-acetyl glutamate, carbamoyl phosphate synthetase 1 cannot work, right? Whereas in this case, the CPS2 cytosolic CPS, N-acetyl glutamate is not required. Once carbamoyl phosphate is formed, then this carbamoyl phosphate, it reacts with the ornithine. It reacts with the ornithine, and carbamoyl portion of carbamoyl phosphate, it binds with the ornithine to give rise citrulline. And this phosphate is released as an inorganic phosphate. This particular reaction is catalyzed by the enzyme ornithine transcarbamylase. So, enzyme is ornithine transcarbamylase. This is also a ligase class of enzyme. You might be thinking, what is ornithine and what is citrulline? Remember, ornithine and citrulline both are amino acid, but these both amino acid, they are never found in the protein because there is no any genetic code which code for this ornithine or citrulline. So, we can consider ornithine and citrulline both as a non-standard amino acids, and in addition to that, both of these amino acid, they are the basic amino acid. Now, once citrulline is formed, citrulline can transport across this mitochondrial membrane, and there are special transporters are there in the mitochondrial membrane which transport citrulline from inside the mitochondria to the outside. That means now citrulline, it can come into the cytosol. Once citrulline is inside the cytoplasm, it reacts with aspartate. It reacts with the aspartate and give rise to arginosuccinate. Now, this reaction, it also requires energy. So, here ATP is used, but here instead of 1, 2 high energy phosphate bonds are broken down. So, ATP gets converted to AMP and 2 high energy phosphate bonds are broken down. So, pyrophosphate is released. So, this energy is equivalent to 2 ATP, and this reaction is catalyzed by the arginosuccinate synthetase. It is arginosuccinate synthetase, and this is also ligase class of enzyme. Here aspartate is providing second amino group C. Here free ammonia is providing first amino group of urea, whereas this aspartate is donating its second amino group to the urea. Once arginosuccinate is formed, arginosuccinate goes under further reaction, basically it undergoes breaking down reaction, and it's broken down into two parts. One is arginine, which is standard amino acid, and second one is the fumarate. This breaking down reaction is catalyzed by the enzyme arginosuccinase. Sometimes it is also referred as a arginosuccinate lyase, but both points to same enzyme. This is of the lyase class of enzyme. Here this arginine is the immediate precursor of the urea. See, arginine in the next step, it is acted upon by the enzyme arginase 1, and what this enzyme will do, it breaks down arginine into ornithine and urea. So, here urea is released. This arginase 1 is of the hydrolase class of enzyme. It is of the class hydrolase. That means it adds up water molecule to arginine and breaks down it to urea and ornithine. So, to complete this reaction, we have to add water molecule into this. So, now urea is generated, but what will happen to this ornithine? And before that, one more important thing, that this arginase 1, it is exclusively found in liver only. So, that means liver is the only organ which is capable of breaking down arginine to ornithine and urea. For other organs, they cannot break down arginine to urea and ornithine, and that is the reason urea cycle occurs only in the liver. Now, similar named enzyme, it also occurs in the kidney. In the kidney, the similar named enzyme is the arginase 2. But in the kidney, the role of arginase 2 is not for the production of urea. Rather, arginase 2 in the kidney, it controls the availability of arginine for the synthesis of nitric oxide. So, their role in the kidney is entirely different. This thing we have to keep in mind. Now, let us come back to urea cycle. What will happen to this ornithine? This ornithine, it will be again transported inside this mitochondria, and there are special transporters available on the mitochondrial membrane which transport ornithine from the cytosol to the mitochondria. So, here as we can see, ornithine is regenerated and this cycle goes on and it produces urea. If we look at the energy consumption, as you can see that 2 ATPs are utilized over here and 1 ATP, but here which is equivalent to 2 ATP. So, total 4 ATPs, or you can say 4 high energy phosphate bonds are used for the synthesis of one molecule of urea, right. So, we can say that urea cycle is quite energetically expensive process. Now, this urea cycle is interestingly connected to the TCA cycle. See, inside the mitochondria, there is one more important cycle going on, that is TCA cycle or Krebs cycle, right. In the TCA cycle, we know that the starting material is the oxaloacetate. This oxaloacetate, it undergoes multiple cyclical reaction which ultimately get converted to fumarate. This fumarate then gets converted to the malate, and malate then able to regenerate oxaloacetate. So, as you can see that in the urea cycle, ornithine is regenerated, and in the TCA cycle, oxaloacetate is also regenerated. This is the similar looking mechanism. Now, interesting steps of the TCA cycle which are in connection with the urea cycle are this fumarate to malate and malate to oxaloacetate. See, fumarate to malate, this conversion is catalyzed by the enzyme fumarase, whereas malate to oxaloacetate, this conversion is carried out by the malate dehydrogenase. And during this action of malate dehydrogenase, one molecule of NAD plus is reduced to NADH. Now, this NADH, it can generate 2.5 ATPs. So, 2.5 ATPs can be generated by this NADH. Now you might be thinking that how this TCA cycle is connected with this urea cycle. So the answer is that here, as you can see, fumarate is generated, and this fumarate is also participating in the TCA cycle. But the problem is that this fumarate is the cytosolic, whereas the TCA cycle fumarate is the intramitochondrial, and fumarate cannot cross this mitochondrial membrane. So what happens? Inside the cytosol, there is one isoenzyme of this fumarase, of this same enzyme, which carries out the same action but in the cytosol. So fumarase, it converts fumarate to the malate. Then this malate can easily cross this mitochondrial membrane. There are separate transporter for the transport of malates. So now malate is transported from the cytosol to this mitochondria, and now this malate, by the help of malate dehydrogenase, can be converted to oxaloacetate. This oxaloacetate, it can undergo transamination reaction with the glutamates. So with the glutamate, oxaloacetate undergoes transamination reaction. Glutamate gives its amino group to the oxaloacetate, and glutamate gets converted to alpha ketoglutarate, whereas oxaloacetate, it gets converted to aspartate. This transamination reaction is catalyzed by the aspartate transaminase that we had seen in one of my previous video, right? Now once this aspartate is formed, what will happen? Aspartate, it can easily cross this mitochondrial membrane, and it can be reutilized in this place. So it can condense with this citrulline and can give rise to arginosuccinate. Here also there are separate transporter for the transport of aspartate. So as you can see that this urea cycle is connected with this TCA cycle, and because of this reason, sometime urea cycle is also referred as a urea bicycle or bicycle. Why? Because here the two cycles are connected, or as we can see that aspartate is shunted from the TCA cycle to here, and arginosuccinate is being shunted from the urea cycle to the TCA cycle. So because of that reason, this particular connecting link, it is also called as a aspartate arginosuccinate shunt. Arginosuccinate shunt. Remember, urea bicycle, why it is called? Because urea cycle and TCA cycle both are connected. What is aspartate arginosuccinate shunt? It is this linking, this aspartate's link with TCA and this fumarate's link with this TCA cycle. That linking is called as a aspartate arginosuccinate shunt. Now what happens? See, this fumarase and malate dehydrogenase, both of these enzymes isoforms are also found in the cytosol, and one of the isoform we had seen that fumarase is over here. Similarly, malate dehydrogenase isoform is also found in the cytosol. So what happens? This malate, instead of going into the mitochondria, it can get converted to oxaloacetate by the enzyme malate dehydrogenase, which is also found in the cytosol, and gets converted to oxaloacetate. And oxaloacetate, it is very good starting material for the gluconeogenesis, so it can be converted to glucose. Earlier I told you that urea cycle is an expensive process. It is utilizing 4 ATPs, but you can look at this thing differently. See, urea cycle, by connecting with the TCA cycle, it is also helping in the production of this reduced NADH, and so 2.5 ATPs are generated. So actually speaking, 4 ATPs are directly used, 4 high energy bonds are directly used, but 2.5 ATPs are generated. So essentially only 1.5 ATP is used, but still it is quite expensive process, because for generation of each one urea molecule, we are utilizing net 1.5 ATPs. Now this entire reaction, if we want to write in the simplified manner, we can see that in the urea, one free ammonia is used, then bicarbonate is used, both are used in the first step. The second ammonia group is coming from the aspartate. So these things are used, which are ultimately generating urea. And along with urea, this fumarate is also released. So, let us mention that fumarate, right. And meanwhile, as we can see that 3 molecules of ATP are used, one over here, second over here, and third ATP molecule over here, and we have to mention all these 3 ATPs. So, here 3 ATPs are used, but all 3 ATP, they are used differently. See, this ATP, it is providing energy to this step, so ADP plus PI, whereas this ATP it is utilized as a substrate. So, this ATP is providing phosphate to carbonyl phosphate, and along with that energy is also supplied, and this phosphate is removed in immediately next step. So, both of ATP, they are converted to ADP and inorganic phosphate. So, out of these 3 ATP, we are breaking down 2 ATP in the form of 2 ADP and 2 inorganic phosphate. But about this third ATP, this ATP is converted to AMP plus pyrophosphate. So, we get 1 molecule of adenosine monophosphate and 1 molecule of pyrophosphate, right. And along with that, 1 water molecule is added. So, we have to add it over here. So, entire urea cycle can be mentioned by this simple one-step reaction. Now, regarding the energy uses, we can say that 4 high energy phosphate are used. 4 high energy phosphate are used in the urea cycle. But if we talk about net or direct ATP uses, then we can say 3 ATPs are used. But we have to keep in mind that 2.5 ATPs are being regenerated by its linking with the TCA cycle. So, I hope that everything about the urea cycle is clear. In the next video tutorial, I will discuss about the regulation of the urea cycle. Thank you.